Hello all. So this week we are looking at cellular respiration and fermentation in lab. It's a lighter week this week, and this content is content you may have already heard in lecture or be working on in lecture currently. So you might have a little overlap. You might be hearing it in lab first and lecture second, or lecture first and lab second. But basically these two processes, so cellular respiration and fermentation, are basically the idea of breaking carbohydrates down to create energy for our cells. And so the energy that our cells like to use obviously is ATP, adenosine triphosphate. Now, it does this best by breaking down simple carbohydrates like glucose, fructose, sometimes lactose, sometimes sucrose, which is actually more of a disaccharide. However, that doesn't mean that your diet needs to consist of just carbohydrates, because our body is really good at figuring out how to do this process of taking proteins and fats and breaking them down into simple sugars. It's actually highly advisable that you do not eat a lot of carbohydrates or sugars, because that is actually not good for our body. And there's a lot more research coming out about a lot of autoimmune diseases and different diseases in our bodies, inflammation in our body and such, that is actually being linked to sugar, as well as a huge rise of diabetes too. So let's look at the cellular respiration is where we're going to start reaction. So if we're looking at this, I'm just going to pull up my pen real quick. Everything on the left side of our equation, so the left side of this arrow, okay, again, I'm kind of going through this. So this arrow, which is pointing to the right, are our reactants. I wrote reaction there. Let me try that again. Reactants. Um, That's better. So that's not right. And everything to the right of the arrow will be considered products. So our products are hanging out over here. So if we look at this, our reactants are glucose, and that just means that there's one. There's no number before it. There's one glucose. And that means it's added to our other reactant, which is oxygen. But in here, in this case, we have 6 oxygen molecules. So these two things are our reactants. On the right side of the arrow are our products. So here is carbon dioxide, CO2. There are 6 molecules of carbon dioxide. Here's water molecules, and again, there are 6 water molecules. And then here's our energy, again, in the form of ATP molecules, okay? And the amount of ATP molecules that we have will change depending on what happens inside the cell. So again, this is happening inside the cell structure. Now, so let's look at this real quick. First of all, this is called a balanced equation, and I may have gone through this with you guys before, but let's go through it again. The equation is balanced. That means that there are equal amounts of atoms on either side of the equation. So here's my glucose molecule. From my glucose molecule and my reactants, how many carbon do I have? Well, if I look real quick, I can see right off the bat I have 6 of them because my subscript tells me so. How many hydrogens do I have? Well, I have 12. So again, that's just what's telling me in my molecule. I have 12 atoms of hydrogen. And then again, when I look at my oxygen for glucose, I have 6. So that encompasses all of my atoms that make up that one glucose molecule. However, that's not the only reactant I have. The other reactant I have is oxygen. Now, if I take this by itself, this is kind of what it looks like. This is not the bonding that would actually occur, but it's an oxygen bonded to another oxygen. So that's why we have this molecule. So here we have 2 oxygen molecules, or atoms, I should say, bonded to each other that create that one molecule. But I have 6 of these, so I really would have to write out 6. So on and so forth. I'm only going to write out 3, but I would have 3 more. And so when I look at this, I'm actually multiplying my molecule number by my subscript, which means that I have 12 oxygen molecules, really, not molecules, excuse me, 12 atoms in this molecule. So I would have to do this, and I would add these together, and on my reactant side, I would actually have 18 oxygen, okay? So I have 6 carbon, 12 hydrogen, 18 oxygen. Now on the other side of my equation, I have my products. So let's look at those because we want to make sure that we are balanced again. So here we have carbon dioxide. If I separate this molecule out and I look at just the atoms, I have 6 carbon right here. Okay, and then I have oxygen, that oxygen molecule again, now that when I separate it, so that would be the O, and then I have 6 of them again. So in this scenario, right here, I would have 12 oxygen. Okay, now if I come back over here to this wonderful one right here, my water molecule, and I separate it, I know that hydrogen has a subscript of 2, so we have basically 6 of these tiny little molecules. 6 times 2 is telling me I have 12 individual hydrogen atoms. So here's my 12. But then again, I still have this oxygen hanging out here, and oxygen doesn't have anything attached. It's not an O2, it's just an O, but I have 6 of them. So here again, I have my 6. And if I add them together on this side of my products, I have 18 oxygen. So if I look on both sides of my equation, I want to make sure I'm balanced. And I know I am balanced because here I have 6 carbon on this side, 6 carbon on this side, 12 hydrogen on this side, 12 hydrogen on this side, 18 oxygen over here, and 18 oxygen over here. So this is what we consider a balanced equation. Now I'm going to go to the next screen and then I'll come back to this one and look at what's actually happening with the reaction. So in the reaction of cellular respiration, we have what we call oxidation, and we also have a reduction reaction going on too. So oxidation removes, so it takes away an electron. Remember, electron has a negative balance to it, a negative charge to it, and the electron is usually always going to follow a hydrogen ion, and hydrogen ions by itself are positive. And so when this hydrogen leaves, the electron goes with it. Okay, and so again, they're going to balance each other out and become neutral wherever they go. Now, the oxidation actually removes the electron specifically from the glucose, so the C6H12 O6 molecule, okay? Now, the reduction is also happening in the same reaction as well, but reduction actually adds electrons. So that's kind of a funny thing, because usually when we think of reduction or reduce, we are taking something away. In this scenario, you've got to get in your head, it's completely opposite. So reduction means adding an electron, again, negative, and that electron is going to follow still that hydrogen atom. And again, this is being added to, the water molecule. So if we go back to that last picture of our wonderful overall equation, I'm just going to change my pen color here. We can see this is happening. So in this scenario, oxidation, like I said, is removing. And what happens is if what we're going to do is take out of the glucose the hydrogen. So all these 12 hydrogens leave, okay? And then again, what's following them are 12 electrons then, okay? So these guys are going to leave the reactant side. They're going to go all the way over to the other side, right? So they've left. And when we leave, get rid of this, what are left? Well, I'm left with carbon and oxygen. And I have 12 and 6 and 6, excuse me. So when I look at my product side, here is the results of that oxidation is that I have carbon dioxide molecules and I happen to have 6 carbon dioxide molecules, okay? Now at the same time, when we talk about the oxygen reactant here, so here's just the oxygen, we have 6 of them. This is reduction. Reduction means, again, adding to. And so what happens is we have these hydrogens hanging out, 12 of them hanging around, and we also have 12 electrons hanging around. They need to do something. They need a job. They're not going to just hang out by themselves. So what ends up happening is we do reduction. We add them, so they're going to come all the way over here in the products, and they're going to be added to that oxygen in the form of H2O. Again, we have 12. So here we didn't 12 12 still. So we changed the molecule. So instead of it being oxygen here, it now becomes water. And so this is the reduction process. So it's basically removing or adding a hydrogen ion, which is positive, and with that positive ion removes an electron. So again, you'll see that none of these molecules have charges to them because they're balanced. Remember, for every positive polarity, you should have a negative polarity, and therefore that is neutral. Okay, so this is the oxidation reduction reaction that's happening within cellular respiration. So let's just take a quick breakdown before our video ends and move on to the next one. And we talk about glucose. So we know that is the simple sugar that our body wants to use to make energy. And so this is either happening in the mitochondria or near the mitochondria of the cell. Remember, mitochondria are those small kind of kidney bean shaped organelles within the cell. There are many of them hanging around. So depending on what's happening, what stage we're at. So it depends on, again, what is happening in this process for things to happen. So basically there are some stages here. So we have glycolysis. Okay, so glycolysis, lyse, to cut, to split the glucose. And so this is our first phase. And this is going to happen in the cytoplasm of the cell. So it doesn't happen in the mitochondria. Glycolysis is basically in the cytoplasm right near the mitochondria. And the job is to take the glucose and split it, like I'm saying, lysing. And what we get is a new molecule that we call pyruvate. We actually get 2 of them. Okay, now depends on what's happening here. Depends on if there's oxygen available. So if there's oxygen available, we're going to continue with cellular respiration. Okay, if there is no oxygen, what's going to happen is basically we're going to continue in the cytoplasm and we're going to move into that fermentation process. So we'll talk about fermentation at the end of the PowerPoint today. But so we are going to hopefully move, because there's oxygen present, into cellular respiration, which would then bring us into the mitochondria. So that's when things happen, depending on oxygen or no oxygen. And you'll see there's going to be a lot of electrons floating around. You're going to see these coenzymes that we call NADH and FADH2. You'll also see them as NAD. So the hydrogen ions have been removed, and you'll also see FAD, hydrogen ions removed. There is a kind of secondary preparatory stage, which we kind of will talk about. It does have a third stage, which we call the citric acid, or also known as the Krebs cycle. I'm just going to write it off to the side over here. So Krebs cycle, or you could call it citric acid. It's the same thing, whichever one you want to learn. And then the fourth kind of final stage is what we call the electron transport chain. I just put ETC at the moment over here. And this has to also do with the ATP synthase and creating our almighty wonderful ATP, because that's what we're doing here. That's the process. Okay. So first step. Glycolysis, like I said, the job is to split the glucose, just like the name implies. Lyse means to cut or split. What are we cutting and splitting? The the glucose, okay? And we're going to have to, in this process, like I said, remove electrons, and those electrons are removed from the NAD. You'll see this has a positive charge to it. And the end result of this stage is that we split that glucose and it turns into what we call pyruvate. And now we have 2 molecules of pyruvate. So one glucose, right? One glucose gets split, and therefore we have 2 new molecules of the pyruvate. Okay. And what's going to happen next depends on whether there's oxygen present or no oxygen present. So I will begin the next video looking at oxygen versus